Thanks, Pauline, for that very generous introduction. I hope there's time left for the lecture after that. So it's my great pleasure to introduce Professor Monique Simmonds to deliver her lecture. Monique started out her scientific career at the University of Leeds and then moved on to Birkbeck College, where we overlapped briefly all those years ago, working on some areas of insect anti-feeding. So it's a real pleasure to introduce her this evening. Monique is fellow of a number of learned societies, the Royal Society of Entomology, the World Innovation Foundation. She's a fellow of the Society of Biology and also the Linnean Society, and is also editor on a number of journals, including editor-in-chief on the Biochemical Systematics and Ecology Journal. She's a visiting professor at Birkbeck College, the University of Greenwich, and also the School of Pharmacy at the University of London. Following her PhD at the Birkbeck College, then Monique joined Kew Gardens, where she has remained, rising to the level of Director of the Kew Innovation Unit and Deputy Keeper and Head of Sustainable Uses, the Plants Group. Her interests are wide and varied. They include looking at role plant-derived compounds play in plant-animal interactions, coming up with chemical authentication methods for a variety of agencies to make sure that the quality of plant-derived products are appropriate, and also looking very much at the details of biological activity and chemical structures of extracts and isolated compounds from plants and fungi. And I guess it's that area where the talk will focus this evening. The talk, as Pauline has mentioned, sponsored by GSK. It looks into the research into the economic uses of plants and fungi, their potential as cosmetics, pharmaceutical and agrochemical leads, an area that's dear to many of the hearts of medicinal chemists, and also sources as sustainable harvested medicines. So I'd like to invite Monique to present a lecture that's entitled Plants and Health from Garden to Drug. Monique.
Thank you very much, and thank you for inviting me to tell you a little bit about the work that's going on at Kew in this area. I'm really going to concentrate on some of the work that's going on medicinal plants, but it's informed, and I think by other areas of research, and I'll hope you see those links. Why do we do it at Kew? It's often a question. We're not a pharmacy department. We're a conservation institute, but the reason in many areas of the world is that people rely on plants as form as drugs. There are big issues that we face at the moment with changes in land use and the need for kind of prioritization of plants that can be conserved. And to support conservation, having a use of the plant is very important. So a lot of our projects are aimed at looking at the science behind folklore. So it's looking at traditional knowledge. Can we explain that traditional knowledge, especially associated with medicinal plants? And I hope you'll see that theme through the talk and how we can use that type of information to get us from just an idea about a folklore, you could say, a remedy, to a potential drug. It doesn't always go that way, but it's clear in some cases it does. There's quite a lot of literature out there about the role of natural products in drug development, and it's estimated about 4 of the drugs that we currently use come from natural products, not always directly derived, but synthetic routes. That's not just plants. Fungi, huge contributor, and also bacteria, and more from on the sea as well. But nature is incredible with the diversity of chemicals. It can evolve, and evolving there because there is a need often to allow, say, a plant to survive in a certain area. And I think that's a key to some of our discovery programs. Why is that compound in that plant? So the work that I'm going to describe, and as the basis of this lecture, is mainly done by my research group in collaboration with a lot of other people. So where organic chemists, neurobiologists, entomologists, pharmacists, etc. So at Kew there is a group of 25 people that are non-botanists that are behind what you're going to see today. Taking on board the expertise from the taxonomists, the horticultural staff at Kew, plus the lawyers. And we couldn't do this work without lawyers and agreements, and I think you'll see why, because the world here has changed a lot. And of course our collaborators and funders. I should add that the group that I have at Kew is on self-funding, so it's research grants and links with industry that we rely on to support the work that I'm going to present. So the basis for our work, and here the stars indicate the areas that I'm really going to kind of mention in the talk, is the use of traditional knowledge and the evaluation of activity associated with that traditional knowledge. Active compounds involved in activity-guided fractionation work that we do to help identify the active ingredients in those plants, and that's changed a lot. Drug discovery, and that can often be based on the ecology of plants. Why are those compounds in their plants? But I'm not going to deal with so much of that, but I will give you examples. Authentication of plants used in medicines. That's an area we are getting more and more involved in. Every year, over the last 10 years at Kew, we've been dealing with about 1,500 to 1,750 extracts that are coming into the trade that are sent to Kew, or we acquire from, say, internet, or from going into the markets, looking at those, are those extracts or those plant-based medicines, do they contain what they say they do on the packet? And about 35% of those that we're looking at are, so to speak, not fit for purpose. It could be that it's the wrong species, and it could be that it's the right species, but it's an extract that doesn't contain the active ingredients, or what are thought to be the active ingredients. In an age where increasing interest in medicinal plants, this is an issue. Now, does it matter? Yes, it does, because it undermines the use of medicinal plants. We need to have better quality control in this area. Now, there's an increased interest in naturals, but there is a decreasing potential to identify plants. Many of you are going to university now to be able to be taught taxonomy at a university. Very few university courses. It's something that's been lost at school. And yet, we're in a world where natural alternative therapies, etc., are gaining more interest. So I think that's often underlined some of the issues here about not knowing exactly what the plant is that you're putting into a product. You need to have somebody helping you confirm it. And this is a really important area when you're identifying new plants that could be possibly coming into the trade. Have they correctly been identified? So that's what's underlying the authentication of plants. The sustainable supply of medicinal plants is important, as I indicated, with changes in land use. Often for food and fuels, etc. Have you got that supply chain? And that's when you get adulterants coming in. And then the conservation of the medicinal plants, identifying those that really have a potential use to communities. And then the names of medicinal plants assisting. And I'm going to give you one or two examples where that really is important. Now, I mentioned lawyers, and they've had a huge impact on assisting us do this work since 1992. Now, the Convention of Biodiversity was part of a World Summit that took place in Rio de Janeiro, which gave really the sovereign rights of people, of countries, to the genetic resources in those countries. So gone were the days where you could go in and do, a trip and pick up some bacteria, plant samples, etc., and then come back to your university or to Kew and do research on them and find something and then just take it to market. You need to go back and have agreements with the countries, and that really has changed a lot of natural product kind of discovery work. It's very difficult sometimes to get those agreements. Who do you get the agreements from? That's just the resources. Getting the plant, you need to have the right permits. If you put then the complexity of traditional knowledge on that, it becomes more difficult. And Nagoya, which was one of the World Summits that took place in Japan in 2010, which was very well attended. So you're talking about, these are international treaties that have been, in this case, 193 parties taking place. And looking here at some of the issues associated with the development of drugs and use of biodiversity. So here you have new access and benefit sharing policies, which include the use of traditional knowledge. So if you're using traditional knowledge, you need to make sure that somehow there is a link back to the owners of that traditional knowledge. Not always easy to do. And then the other side of that, you get TEEP, which is to look at the economics of ecosystems and biodiversity. And that's putting a value on some of the plants. So you've got two aspects here going on. One's kind of a huge opportunity of trying to show value of plants, and others is kind of the complexity of getting into some areas with the right permits. And it's very difficult to know from the outside whether you've got the right permits to do some of this work. Although it's actually getting much better than it was in the 1990s. The other framework by which we work here at Kew, and it's again linked up with some of the decisions that have been made at the World Summit, is a global strategy for plant conservation. Now, there was a meeting in Dublin in 2010 that put forward the new targets that to be delivered by 2020. So a lot of the work we do, and a lot of it you'll see is in collaboration with others, are really looking at how we can assist with our medicinal plant research, help with the conservation, therefore, of plants, and looking, therefore, at the trade of some plants. So you've got a decrease, therefore, in the wild flora that could be potentially endangered by identifying something that's very active. But you've got all wild harvested plant-based products sourced sustainably, so you can actually record the numbers, so you can put some facts. And then this last one is the indigenous local knowledge, innovations and practices associated with the plant resources maintained or increased, i.e. giving value to that traditional knowledge that had been acquired by those communities. And that knowledge is not lost. And therefore, these help support those communities, but also to outside people as well. So we're working very much within the context of the Global Strategy for Plant Conservation. So let's kind of go through some of that's the policies. Let's get now onto the research. So welcome to the pharmacy. This is what you could see in parts of Cameroon. If you've gone to parts of Africa, and the same in parts of Asia and South America, you will come across stalls like this. And what are in those jars at the back? Person who's going to sell them to you, kind of potions, etc., and so on. Are they good quality? Do they have an effect? What's the use of these? Are these symbolic? When you start to look at some of these, people keep on coming back for them. Now, is that just because it's, they think they're getting something back? When you start to look at these in a little bit more detail, it is quite fascinating. And I think one of the most fascinating things that in some of the work that we've been doing, and others, if you look at literature, if you look at the arrows, the knowledge that's gone in to knowing exactly the right amount of a toxin you put onto an arrow that will knock down the prey but doesn't taint the meat. Now, over the years, the experimentation that went on to be able to do that, we haven't got enough sometimes of that background information. Of course, there's an interest in the arrows that are knocked down because of the potential of drugs. So there's a lot that can go behind that, and a lot of people think, That's all unknown. But then, if you look at what's happening in China, and I'm going to give some other examples, there you've got, thousands of years of knowledge and selection of those plants, and more and more interest in traditional Chinese medicines in the West. So how do we capture that? We face issues here because it's in Chinese. It's often difficult to get to some of the information about. It's a different form of how they use the plants as well, compared to our westernized system. So there are lots of challenges. Now, if you go to our own pharmacy, especially prior to 1948, the development of the National Health Service, this would have been in the cupboards in the pharmacist stores, etc. That would have been your quality control standards that the pharmacist would have used before giving you some of the potions or the drugs, etc. If you look at some of these cabinets, which contain from 300 to about 400 different samples, and there's 150 species that are about common to a few of these cabinets that we've investigated. We actually only know the chemistry, about 75 of those 150. So, things that are not all that distance from our use. So we still need to know far more about what those active ingredients are. Because it's not all that long ago that these were the main form of our medicines, our treatments, etc. So when you compare this, which we think, okay, we know far more about what we were doing compared to what that store in Africa, actually, we can't be too complacent on our lack of knowledge about our own traditional medicines. So within Kew, we've got a range of projects, and the ones that are kind of highlighted in red are the ones I'm going to mention today. So we're working with over 800 organisations in 100 countries on different aspects of this work, to different levels of kind of capacity. In some cases, it's just screening the plants, identifying the plants that are being used. Others, it's more active research programs. So there's very much an international array of projects. So I'm going to just illustrate a few of the projects here. This is a project that started about 2002-03 in South Africa. We were invited to take part of this project by Umtati and a UK NGO, Garden Africa. The reason we were invited is because of support in identifying some of the plants, but support in helping to do some of the chemistry on the plants. Now, these were plants that were used by communities there that were, well, the traditional healers there that were supplying plants to the communities in areas where there was not really access to westernized drugs, especially for the treatment of HIV and related conditions, things like TB. So we got meetings together with some of the traditional healers, and the lady here in the middle, Phyllis, is one of the kind of coordinators of the traditional healers in the area we were working with. They identified the plants that they had an interest in, that they hadn't got access to. Now, Grahamstown is on the garden route, and the garden route, as it's developed over the years in South Africa, has attracted other forms of activities like the game parks. And the game parks with the animals, etc., because of the fences, etc., that have come up in that area, the communities don't have access to the land that they were collecting their plants from. So they aren't able to acquire some of the plants that they need for their patients. So we were able to get some money out of a Darwin Initiative grant, UK government as well as EU funding, to buy some land that they could use to grow the plants. And here we have an example of of some of the plants being grown by the communities. And then we have exchange of information about the plants. Now, we identified 45 species that they had a use of. And there was discussions about how you transfer those plants from one area to another. And this is sometimes the challenges you face in these community-based projects, because it was very much a science-driven project. How do you get the plants cultivated, etc., so you can supply? When we purchased the land, they said, fine, thank you very much, but we need to have the spirits in that land. Okay, how do we bring the spirits? We need to bless it. Fine, we have a blessing ceremony. We need to bring those spirits with the plants. Okay, how do we do that? Well, you need to take the soil. And you're kind of thinking, why didn't I think of that? Because it's pretty obvious, isn't it? It's bringing the nutrients and because you change plants from one area to another, you can affect their chemistry. Now, they don't always use the same language, but they do know that some plants from one area are better than from another. And in some of the work that we've done now, we've taken some of those plants and we've taken a batch of those plants, kept them in the area, taken one batch and we've just transferred them into the soil that we would have. Another batch, we have taken some of that soil with those plants and we've looked at the chemistry. And in 5 species, there was a huge difference in the chemistry. What we could therefore have done is to propagate material that actually would have not had the same chemistry. Now, would have had the active ingredients. That's another story, and I don't want to get into that area. But I think over the years, this is a common mistake that has been made. Not enough attention has been paid to making sure that active plants are those that are propagated and supplied. And therefore you get, okay, this is no wives' tale, it doesn't have any good, doesn't have the right properties. So more care, and especially when you're doing this type of project with kind of vulnerable groups, and you go in as kind of an authority, and you can end up actually giving very inaccurate kind of information. Right plants being propagated, but actually inferior plants therefore being supplied to their patients. So it's a huge learning curve for us in this type of work. So we can take that model, and this is Namibia, where you can work with communities, again identifying plants, in this case it was food plants as well as medicinal plants, and create an area like this in only a few years to an area like that, with therefore a supply chain for those communities. And the trick here is what we're trying to do is locally appropriate plants. So it's not always our westernized plants that they're growing. These are African plants with some of the properties. There are a few in here that actually aren't, but in the majority of cases they are. And they can do different kind of companion planting and things like that, so that don't have the expense of some of the insecticides, etc. There's a lot more, though, that can be done in this type of project, working with, again, other groups that sometimes have got more experience in this than we have. So that's examples of some of the work, therefore, we're doing on medicinal plants and conservation with communities. Another is on specific disease areas. And Kew's had a long kind of tradition on antimalarial plants. So I'd like to kind of share with you where we are in this project, and again, how we use historical information and new information into assisting us identify which of the plants we think are worthy of further study. So we go back in time and look at Sakona with quinine, a main source, and go back to the exports that took place in Europe back in the 1632, and kind of the English remedy, although it was actually the Portuguese and the French who developed it first. But we actually, within England, put a lot of the science into the development of these antimalarial drugs. And it became popular in Britain after the 1670s for treating fever. Remember, we used to have malaria here. You notice, of course, it doesn't say anything about malaria. It was treating fever, because it was in the 1940s that they identified Plasmodium and the link with the fevers and the causal organisms. And what's nice here is the kind of association with some of the kind of history of Britain and Sir Hans Sloane and the Physic Garden in Chelsea. He did a lot of travelling around the world, bringing a lot of the medicines that we have into UK. And then some of the work he was doing in Jamaica when he studied the fever plant. Now, he didn't use it so much for fever. He was using it as a eye ointment. So it's kind of parallel things that go on in some of them are dropped because more actives were found. And it's a very interesting history looking at how he developed and developed the different areas and the uses of Secona. Then this comes to Kew, and this is where it's fascinating, the collections we have at Kew. We've actually got some of those, not the initial collections back in the 1670s, but 1820, these Seconas that were used to develop some of the drugs that were marketed in Europe in the 19th century. So we can go back and look at those samples. We can therefore look at the differences in chemistry in old samples compared to now. Because plants do vary over time, so we can go back and see, is the Secona chemistry the same now as it was in the past? And actually, when they were selecting plants, this is where Kew did play a role, because plants used to come in from different parts of the world through Kew and then into parts of the empire for developing the plantations, etc. And of course, some of that material was selected, which was not viable. So the same mistake that we could have potentially made in that other project in South Africa was going on in the development of Secona. And sometimes you can go back and you can see the inactive plants that we were actually trying to grow in parts of India. So we can look at the development there for the plant material to those samples. And this is part of the economic botany collections here at Kew. So that's Secona, and then you can look at what's happening now in Artemisia, which is it's a traditional Chinese medicine, and isolated here, artemisinin, which is the active ingredient, back in 1972. And a lot of funding from WHO and other organisations has gone into the development of artemisinin as an antimalarial drug. So here, both of these are based on traditional knowledge about the use of these plants in the treatment of fever. Now, when we look at our literature on antimalarial plants, and we've got information about 3,000 different species that are used, let's say they're used for treating fever. Sometimes an assumption that antimalarial, sometimes we have got more information. Now, we could list those plants from A to Z, but we can also take or make use of the molecular data that is available of the flowering plants. Now, the molecular data has been used to look at plant relationships. So it's looking at families of plants. Are they related or not? So here we have a genetic kind of profile of all the estimated about 300,000, 400,000 of the plant species that are out there. And these are the different orders. So there's your rose plants would come in here, your mints, etc. are there, your potatoes would fall in here. And what we've done is we've superimposed where these arrows are, the plants that are used as antimalarials, and the ones that are kind of bolder are the ones where there is better evidence. Now, how do we use that information? One, we can see that it's not randomly distributed. We can use this information because if those plants are related, there's a high chance they will have similar biosynthetic pathways on their chemistry. Now, if we've got a plant here, which in this group here we already know some of the active ingredients and we're looking for novelty, let's therefore not go there, because it's highly likely we will identify a type of chemical that's already been identified. The chances are that we may be better looking at some of the plants here, because these are the ones that haven't traditionally been used for drug development in antimalarial. So you can test hypotheses based on traditional knowledge. Or is it the fact that, because plants do share similar types of chemicals, that we've selected plants over the years because they have a similar type of chemistry, and that's why they're used as anti-malarials. There's a whole lot of hypotheses you can test behind this. And the work that we've done, this is William Milligan, some work he was doing with the Yamahami Indians. This is where we get the information, collecting the information. Nowadays, under permits, and all the traditional knowledge nowadays is kept in country, taking into account the CBD. So it would be published, in this case, in Brazil, in other cases in South America, etc. And then we use that information when it becomes in the public domain. It's a project in Ghana that we've been involved in over the last few years. It's where you've got a large population there, 100% of the population is in risk areas for malaria, and a high level of mortality, unfortunately, in the young children. So we've been working with collaborators in Ghana, finding out about the traditional uses, and we've been working in these four areas that have got different levels of malaria. So high levels here. So when you've got a lower level, are they using, again, it's different geographical distribution, therefore slightly different plants, but actually there's some common plants in these areas. Are they actually using the same plants in these areas? And are they the similar type of plants that are used in different parts of Africa, etc.? So what we did with these four sites, we interviewed over 550 people, and you often get different information depending on how you do those interviews. So you often have to standardize. So sometimes you just talk about the plants, when they're used, and of course they'll be using local names, so you often have to therefore make sure you know what the plant is. Or do you bring some plants together and say, do you use these plants? You often get different information. Male-to-male interactions, you get different information compared to what you do female-female, and it really does depend in some of the cultures. Who are the people who gather the plants for the traditional healer? Because it's not always the traditional healer that will gather the plants. So having knowledge about those communities, who are the treaters, but who are the gatherers of the herbs. And again, you can do controlled studies when you're gathering this information. So this is one of the communities that we were working with in the hippopotamus sanctuary area in northern Ghana. And when you start to bring some of that data, this is just from one of those communities, you can look therefore at the different uses on females and males. You can see from here the high use that is made in these communities of antimalarial plants compared to westernized medicines. And you can also find out a little bit with the effect of education on the selections of the plants here. And again, basic information, a lot of them were using traditional plants. But a little bit you can see here of the westernized medicine as well. Even those that were, fortunately we haven't got many to interview here at this stage who had gone on for university. There was still a few, and some of the communities actually were still quite high, who were still using antimalarial plants. And from that data you can start to identify some of the species that are used, and therefore looking, and it's really important here, to look at how those plants are used. In this case here you've got the dried leaves, so it's the leaves that's used, it's not the bark. Often in Africa there's quite a few medicinal plants as the bark, here it's the leaves. And it's used 3 times a day, so you have an idea of therefore the concentration that's required. that information becomes important when you're doing some work in the labs. It's water extract. It's not ethanol. It's not a solvent that we would use in some areas. Right, you've boiled. Sometimes you need to know a little bit more about what it boiled with, because that's often the key. And therefore the concentration. And we've done quite a few studies on this. It's often very important that you start off with the right, you could say, concoction before you start your activity-guided fractionation, because it's very easy to dismiss something because you started off with a ethanol extract of dried leaf material. This is fresh. The chemistry varies. The other part of the work that we've been doing is when we go into the communities, that's the community base. So what then goes from the communities out there in the field to the markets, etc. And the market areas that we've been looking at, that's where you get an awful lot of substitutes, poor quality material getting in. It looks like there's much better quality in the village communities compared to the material that's been sold in those drugs, you can say, the charlatans, etc., and so on, are getting into some of the market areas, so poor quality material getting into the trade, and therefore larger number of communities, because there's a lot of people buying this material, and that really does undermine the use of these plants for treating malaria. And this is one of the ones that we did find quite consistently was getting from the market, from out in the field to the market, Monodora. Again, here it's never used by itself. It's used in combination with Zingiber ginger and also different pepper species. It's a combination. Again, it's the use. It's the leaves. And we need to know far more about kind of the combinations that are used here and why it's those combinations. And that's most likely affecting uptake in the gut. So how do you mimic some of these when you're doing experiments in a test tube environment? Another thing that's interesting is the use of materials from different parts of the world. Azadirachta indica is a neem tree from India, but it is used quite frequently within parts of Africa for pesticidal use, but also for antimalarial use, which is a use. It's also used in parts of India. So a plant that from a different part of the world comes in and is used in preference to some of the local species. And there's another species which is one of the ones we're actually we're doing more work on this one, the Plexanthus. Now, this plant is used in Africa, but it was one of the plants that was used with when you got people migrating with the slave trade. Slaves when they were taken over to the Americas, they often took Plexanthus with them. It's one of the plants that they took. It's also a species that is now used by the Yanomami Indians in South America. So a community, therefore, has developed, an indigenous community is using a plant from a different community. So somehow, we think they've got it in preference, again, to some of their local plants. So it's kind of the history of this. It's very difficult to actually pinpoint how this evolved. Now, but to us it would indicate that surely this must have something in it that's worth further investigation. Again, it's a plant that's never used by itself. If you test it, you find nothing that's antimalarial. But when you start to look for combinations, that's when you start to have some activity in assays. So, going back to Hans Sloman, the work that he did in on the development, you could say, of quinine, etc. Out of all our leads that we've got today, it'd be very interesting to know how he would select the plants and why. I mean, the plants that he did select, kind of his eye ointment and others, when he had an array. There was obviously some selection there. We don't know fully what it was because it wasn't all documented. But out of those 3,000 species that we've got, we've identified about 20, 30 that were further studied. It'd be actually fascinating to know some of the criteria somebody like Sir Hans Sloane would have used. Let's go to China. Now, you can see a real interest in that. We do a lot of work in China and on traditional Chinese medicines, not just for medicines. There are interest in pet care, cosmetics, functional foods, etc. And there's a lot of money being put into the Chinese government into the protection. They're taking it very seriously. The largest number of new botanic gardens in the world is in China, helping to conserve their genetic resources. You can see here the investment of 183 million in their healthcare, and a large proportion of that is actually associated with some of their traditional plants, taking a westernized approach at the time to drug development. They've got a lot of herbal products there based on about 11,000 different species, of which it's just under 4,000 are in the Chinese pharmacopoeia, those that are used in what we term traditional Chinese medicines. Now, there's some exciting things going on in this area, which I think will have an impact on what's going to happen in other parts, say, of Europe here. In the USA, we had first TCM that's gone through for approval in late 2006, and that was derived from a green tea concoction. And at the moment, we've got Dantonic, which is for cardiovascular disease, that is, as of mid-July 2010, started phase 2 trials. Now, if these go through further into actual approved drugs, I think it's going to open the market in this area. But there are huge challenges. And I'd like to kind of just go through how we're dealing with some of those challenges. This is one of the plants, Dantonic it's Danchan Diawan, which is a species of salvia, so it's your thyme family. So the work that we're doing at Kew is to build a collection of traditional Chinese medicines so we can assist, identify the plants that are coming into the trade. So we're going out to China. This is Chris Leon, who since 1998 has spent a few months of most years working in China, going to the different parts of China, again in collaboration, collecting good quality material, plus also market samples. So we have an idea about the differences in the chemistry, but we can also have samples, be they DNA-based, be they for anatomy, but also for chemistry, to assist identify the plants that are coming into the trade. And this is the flowers of the plant, but it's actually the roots that are used in the medicine. So it's a challenge here because we're not dealing with one single compound. So our previous drug development type programs were often based, so if you take this as the complexity of a plant on the chemistry side, we were looking for that, you can magic bullet, we were looking for that one active ingredient usually, and that's our basis of a lot of our work, so that's a traditional drug discovery. It's clear from the work that's going on the traditional Chinese medicines, you have the complexity of those different plants in the formula. It's not just one. You've got maybe the primary ones that are associated with the activity, but you might have what term the carriers, etc., that might be affecting the ability of those actives to be absorbed through the gut, etc. So what we're doing here is getting into what's termed system biology. You're trying to find out what is that profile of chemicals in this plant that's required for having a good quality plant, but also to identify what the actives are within the body, but not just one compound. That's quite clear. So a few years ago now, 3 years ago, we applied to the European Union in collaboration with it was over 20 other institutes for a EU grant, a network grant, which is good practice in traditional Chinese medicines, because there is a need to be able to look at evidence-based knowledge that's out there to meet the needs of the westernised market. You're going to take some plant, like the, say, extracts from the traditional Chinese medicines. We can't rely on previous knowledge. They've now got to go through the westernised system of drug development that meets that criteria. So the grant was to identify best practice. What's the information that's already out there in the literature, Chinese and also in kind of English or European, westernised, you could say, westernised literature? How do we bring that information together to assist what needs to be done to meet the needs of the Western clinical trials, which is why it's so important looking at what's happening over in the States as the plants go through the drug development. So we have a work package here where people are looking at the quality control, which is the part, this is the bit that Kew is heading, and then we've got people from King's College is very much involved, and then we've got the, this is done in collaboration with China as well, we've got the Belgium's involved in the toxicology aspects. It's all being brought together, going out, collecting that information, so we can look at how we get these drugs to market. So it's a gap analysis. What information needs to be done? And what's therefore the best practice to get these drugs? And the same thing, it's not just traditional Chinese. We've used traditional Chinese medicine here because we can see a market, but it also applies to, you could say, European traditional plant remedies as well. So we're identifying priority areas for future research, very much, as I said, evidence-based. Now, kind of you could say, does it matter? Well, we're interested in looking at that potential, realising that potential of plants and drugs, and if they are going to be perceived by the medical establishment, they need to meet those criteria. Now, there's a lot of interest in Chinese star anise because of its potential use in treating SARS. And it's the basis for some of the drugs that were used for treating SARS. And I'd just like to illustrate the complexity of plant names and why it is so important to get them right. Now, you most likely know about traditional, maybe some of you know about traditional Chinese medicine or the use of things like Chinese star anise, because you might be like curries and things like that. What you don't want to do is to get Japanese star anise, which is actually pretty toxic. You can see here they look similar, but they've got different names. Okay, does it matter as long as you know what the name is? This is the Latin binomial name, Latin binomial name. Okay, and this is important that the correct Latin binomial name is used, because again, it's the legislation, it's what you'd use in a pattern, etc. And you think something like this, which is well studied, there wouldn't be a problem, but just take a look at this. So, star anise, that's Alyssum verum, and hook F is what's termed the authority associated with that name. That's why every Latin binomial name has an authority. It's often associated with a person who actually identified that plant. But you've got synonyms, you've got other names that get into the literature to name that particular plant. You look at the Japanese star anise, Alyssum anisatum, that is the Latin binomial name, and that's the authority. You'll notice that one of the names here for this species is actually the name used, you could say this is the accepted Latin name, for the Japanese. So if you had used this synonym for the star anise which you use in your food, you can see where a confusion could lie. You know which one you've got. It's a so to speak. It's a name that's been used in the literature, but you can see it's confusing, especially if you didn't take into account the authority that somebody just put that name down. Okay, that's an academic question, it might be. It actually gets more complex, but there are different names. Well, does it matter? Is it an academic exercise, this? No. You look at what the legislation is associated with these species. The botanical variety, is significantly recognised as highly poisonous. And it's the same in the American legislation. That is star anise, the one you use in cooking. Right? It is classified by EU legislation as being highly toxic and shouldn't be used. And the same in the FDA. So there's an exercise going on at the moment which is supported by the Wellcome Trust that is looking into the uses of plant names to make sure that they actually have the right one, because you can get legislation which could cause a problem, especially if you're trying to develop a product. And for this particular project, we've got a lot of user groups from the industry that are kind of assisting us, so that again, it's not an academic exercise. This is very much to support the industry and therefore support drug development, because what you don't want to do is to get into the literature information about a plant being toxic when actually it's not, because it's the wrong species, because that can cause a lot of problems when you want to register something. So it's really important. Okay, let's get back to kind of areas of drug development. We've covered parts of China, etc., and some of the work we've been doing is kind of UK's back garden, looking at the potential uses of some of the plants in UK and rosehips. Now, in rosehips, we've identified plants that have kind of antioxidant properties, also use in cosmetics and possibly for skin conditions. So we can look at the traditional rosehip that is grown in our borders, etc. This is going where we can use the collections at Kew. So we can look into one of the cupboards that's in a herbarium. We've got the herbarium sheets here. And we can say, well, we know what the active ingredients we're looking at. And Elaine, who's one of the natural product chemists in the group, you're talking to Renata, who's a taxonomist. So she's basically saying, this is what I'd like to do. Can I please have a few little leaves? That's all I need. Don't want to take the whole of your sample because I know what I'm looking for. Out of the cupboards here. So instead of doing a tour around the world to get samples of rosa, we can actually open the cupboards here and do some sampling to identify actually have we got the species that's got the highest level of the actives that we're interested in. So we can start then to do some work on the chemistry to identify the different components. Sometimes they're not always novel, but in the process of doing that, you actually get some academic papers out of it. And here we've done some 71 species, and identified compounds that weren't previously recorded in those plants. So not always active, but it furthers our knowledge about the chemistry of the plants, as well as identifying sources that have got high concentrations of the actives we're interested in. So now I'd like to kind of end up with some area of research, and this is a GlaxoSmithKline lecture, and really kind of thanks to GlaxoSmithKline quite a few years ago now, 10 years ago, about 15 years ago to 10 years ago, when we were actually doing some work with them on Alzheimer's that started our interest in this area. Disease, of course, of increasing interest in looking at drugs, and looking at partly, we started off actually on a whole range of amyloidaceae alkaloids, those types of compounds, and we still do some of that work, but more recently we've been looking at some of the essential oils, not to treat the disease, but actually it is looking at some of the associated conditions such as agitation. And Melanie Howes here in the team is one of the key people that's involved at Kew in doing the chemistry that's associated with the work, and we're working with others in Newcastle, King's College, etc., who are doing the activity work. So, started with some of the work that we were doing, things like galantamine, which is one of the drugs that's been developed already, but is used from snowdrops, etc., from narcissus plants. And there's a review that's been published last year that kind of looks at other plants, 180 different compounds that have identified that have potential in this area. So nature actually could have some sources of potential leads. But in the area of quality control, we've looked at things like Melissa, sweet balm, salvia species, which is interesting. Salvia, sage, wisdom. There was actually a traditional link in some of the salvias associated with treating mental conditions. There's quite a lot of kind of potential to use traditional knowledge on that group to select the species. But we did a bit of a survey, and Melissa and lavender came up as two potential plants that were worth further investigation. And here again, we're identifying what the potential actives could be. So we're making extracts that contain the active extracts, so the active compounds for use in these trials. The idea in some of these trials was going to be topically applied. So there's scientific data from other studies, not from ones we're doing at Kew, but from others that would indicate some of the receptors that have potential targets, and we know some of the compounds in the extracts we're dealing with have the potential to stimulate those targets, so it's got a rationale behind the science here. In this case associated with agitation. And this particular double-blind, which was research sponsored by the Alzheimer Society, where we were looking at Melissa associated with lavandula, where we take a 10% solution in a formulation. It was given to patients over a 12-week trial, and then the patients there's a series of questionnaires, etc. We had blood samples being taken to see if you could actually identify the actives that were in the lotion. Are they actually getting into the blood system, etc.? All those controls were in place. And after 12 weeks, they were monitored. I'm afraid there was no evidence in this particular case against agitation, but there was a trend for improved quality of life. The patients felt better. So I think it's going back and actually, have we got the right extract? Do we have the right composition? So I think we're onto something here. There's further work that needs to be done. But it's actually identifying how we come up with the right criteria here to identify exactly what is meant by improved quality of life. So, hope in this lecture I've given you some examples of using traditional knowledge, of using some of the taxonomic tools to identify potential leads, and some of the areas, say traditional illnesses like malaria, but also kind of some of the challenges like Alzheimer's, that we think plants do have a role. It's unlocking that role, and with us it's often working with the appropriate people with the pharmacy skills, that we can work with state-of-the-art assays, because we see our role is providing support on the botany and the chemistry, natural product chemistry, working with others that can assist evaluate the extracts as potential leads for drug development, as well as helping us identify and validate some of those traditional uses. Thank you.
Thanks, Professor Simmonds, for a fascinating insight into the components of many of the products from around the world. I guess you're okay to answer a few questions. So, if we could ask if there are any questions from the audience. If I could ask for you to wait for the roving microphones that are coming round and hold the microphone pretty close, as we do need to hear the questions. Thanks.
Hi, thank you very much. That was very interesting. Can you just say something a bit about the legality of...
Sorry, could you just hold the microphone a little bit closer?
Can you just say something about the legality of, if you say isolate something that's promising, you can't really patent it if it's a natural product, can you? So the drug companies are are they interested in pursuing?
Yes, some of them are. You can patent a natural compound if it hasn't been associated with a use. It's not the compound per se you file the patent on, it's associated with its use. So if we identify, the best protection is a new natural product associated with a new use that compound stimulates a receptor in a particular, it could be used for treating a particular disease. Yes, you could file that patent. In European law, you couldn't file the plant for that use, but you could if it's a new compound. Or it's a compound that's not new, but it's a new use. You'd be filing a patent on that particular new use. And that's where the advantage would be for that particular pharmaceutical company. It doesn't mean that it automatically prevents the traditional use of that plant going on. There's a lot of fear about that. Say, commercial companies will come in and they'll identify a plant that has been used traditionally, and therefore that community is no longer going to be able to use that plant. That's not the case.
Perhaps I could ask one. Thinking about the regulatory side of things, do you think there are going to be issues associated with licensing mixtures of traditional Chinese medicines in the Western world?
I think there will be, and that's why we're so interested in what's happening over in the States. Because if they start to get these through the FDA, etc., I think by learning about how they've done that, etc., I think that's going to open the doors to other areas. It's not easy because of that complexity. So most likely we are going to have to identify the profiles of the compounds that are associated with its activity. Because what otherwise are you going to be protecting? And how will you standardize an extract if it's going to be used continually? Because that's a key to its success here.
Yeah, some of the aspects of the complexity only just started to dawn on me as I heard you lecture, because it's not all the different conditions of where you grow it, presumably how old the plant is and all this kind of thing.
But once you've got that and you've identified what that profile of compounds is that's associated with its activity, that's when you can standardize extracts. And therefore you think about your harvesting, etc. And the Chinese, many cases, have cracked that. They know when to collect some of their plants, as do in other areas here. I mean, in development of, say, pesticides, pyrethrins, etc., they know that they have to collect the flowers early in the morning because that's when you get the active ingredients, not late in the evening.
I think there was a question in the middle there.
Sorry. Yes, I'm a medical herbalist working in this country. Using, I work as a medical herbalist in this country, using very traditional extracts, I suppose, ethanolic extracts. I was wondering whether you ever looked into those to tell us what are we actually using, because most of the time we don't know.
Yes, we do some work on kind of plants that are used in UK. We're sometimes asked as kind of collaborative projects to identify. Now we can tell you what's in your extract, but we need to be working with somebody who's evaluating its activity. Because the work that we would do is say, yes, in your extract, you've got the compounds that are associated with its activity, but you actually need to have somebody else who we can work with that says, yes, it does have that particular activity, whatever you're using it for. So this is very much collaborative research. And what we try and do is to get people at kind of the state of the art with some of those assays that can support identifying, but it's expensive research. But what we can do is kind of help standardise those extracts. What you need to do to make sure you've got consistently good quality.
Hello. Can you explain, is it possible to control the compounds that are being produced by the plants by adding minerals to the soil rather than transporting the soil to the area where they're being grown?
Yes, and there's a lot of work that goes on. Artemisinin, what's been grown out for the antimalarial, a lot of work has gone into looking at the genotype, the plant material that has the potential, and then making sure those soil conditions are right. And one of the things we've often neglected, it's not just the minerals, it's often the mycorrhiza fungi that are actually out there capturing the nutrients from the plant, and that's something we've often neglected. We're just realising, actually, over the last 10 years, the importance of plants, to plants, of the fungi in the soil that are actually getting those nutrients there. So it doesn't always work when you transfer plant from one area to another, even if you get some of those nutrients sorted out.
How do you deal with the gradual evolution of plants and over time? Because a plant this year won't be the same in a decade's time necessarily, because it's responding to environment. You say change in atmosphere, for example, in Western developing, and I guess in developing countries that's becoming more of an issue.
Yeah, and kind of the domestication of plants can change their chemistry. That's why it's quite interesting going back to some of the Secona barks that were collected, and because some of the interesting compounds are stable, we can actually go back and see if they're the same. I think that's a very difficult one to really identify, because plants are evolving. That's why if it's going to be used in a drug program, we need to know that profile of active so you can control for changes. It's clear if you look at the potential now for growing, you could say, essential oil producing plants in parts of UK, there's more potential than there were a few years ago because of warmer climates. So yes, you've got potential to transfer plants from one area to another. It's not an easy, there's no one easy. I think I'll answer that one. I think you've got to do some monitoring for your plants.
One over here on the right.
In cases where you've identified plants that appear to have been established or adopted in a different colony or area, are you confident that they were taken over? I mean, in the case of Petranthus that you described, are you confident they were taken over at that time, or is it possible that they could have arrived earlier in earlier waves of migration?
In some cases, we know that they were part of the medicinal portfolio of plant samples, seeds that were taken by some of the slaves. They're documented. I can't tell you whether they were there beforehand. In some cases, we just don't know, but we do know that they came with those communities, and it's highly unlikely that those plants were in some of these areas beforehand. There's very little evidence for that. And if when you start to look at the use then, especially with the Yanomami Indians and things like that, the transfer of plants into certain areas, you can see how that's happened to some extent. But whether there was a group of plants there before the Portuguese came, you can't say the gospel that they weren't there beforehand. No. But plants often are kind of, they've got areas where they are distributed. Very few plants have got a global spread.
On your needle in a haystack slide,
Yeah.
you seem to suggest that there was probably an active molecule and perhaps some others that helped its uptake.
Yeah.
Why is it not that there could be several molecules that together are active but on their own arms?
Oh yes, there's kind of a synergy effect. Yeah, what I was trying to just do in that kind of two slides is to show that we've moved from, in part of the work, we've moved from kind of that traditional activity-guided fractionation to find one active molecule. We know from some of those early studies you get to a stage where the activity would start to decrease as you decoupled that extract. Sometimes a carrier or get synergy amongst those compounds or additive effects. To do those experiments in detail to show additive, synergy is jolly expensive, jolly difficult to do. So that's why I was saying, you know, they seem to be important, how they interact. I think you need to do far more work on doing that. But you know, we do know in some cases, especially on gut experiments, that some of the compounds do help uptake from other experiments that people have done. That's, I think, it's so difficult for us to say a traditional medicine which is used kind of as a kind of an oral uptake maybe 200 years ago is a folklore when we look at that same plants now, because again changes in chemistry is the same one, etc. and so on. But having correct information about its use, and as I was indicating, was it used? Was there anything else in that combination? There's one example that I could just kind of identify that would, I think, answer one or two of your questions about kind of the effect of climate. Gallium is used, and sorry, let's say take figwort. Figwort is used for treating ulcers, leg ulcers, as is gallium. That's a weed. I mean, you know, that's cleavers, etc. And most of you got cleavers in your garden. It's traditionally used nowadays in parts of Norfolk, etc., and in areas of Somerset. When we identified it as something that was worth looking at because we could see how it's been used, we thought just go down to Kew, just get some. I mean, it's there in the veg plots, etc. So we did some work on that, and it wasn't active, so we nearly stopped the project. But we'd seen the evidence of its use, so we thought, okay, let's compare it. So we did a bit of comparison on the chemistry, and it was very different. But we also failed to take into account that it was being used fresh and as a poultice that was applied. We were using freeze-dried material that was kind of sometimes had been in the lab a few days, and it therefore wasn't fresh. What we did in that particular case, we freeze-dried the material at the site after it was collected, which stops then the natural breakdown of those compounds, and then we could do some work which helped us identify some active compounds. So, you know, there's lots of paying attention to traditional use really is important. If we're not just going to say this is old wives' tale, it doesn't work.
Two questions here. Second row first, and then.
Is it likely that a lot of very toxic plants, actually the toxicity masks something that could be very helpful, so you wouldn't have got it as a traditional remedy?
Use of aconite and things like that, some of the toxic plants, which in traditional Chinese medicines, they're prepared in a way that gets rid of the toxic compounds, so it's the other compounds that are associated with the activity. We actually don't know, in that case, what the active ones are. Yes, sometimes we're rejecting things because we know about their toxicity, and there actually could be other active ingredients that are there. But, I mean, those are a real challenge to work on, because if they are going to get into the trade, you've got to be absolutely sure that you've got rid of the toxic, so in preparation, etc. So there's a lot of knowledge. And again, traditional Chinese medicine, They very rarely use unprepared, unprocessed material. They use things like ginger. Licorice is often used in their preparation. You make an extract with water or you make an extract with licorice, you get a very different complexity of chemistry coming out.
I don't really understand how your star anise and your Japanese star anise have ended up with so many different binomial names. How has that happened?
Because you've identified it, right? Saying, X number of years ago, you've identified it, and you've given it a Latin binomial name. Somebody in this row has also identified it, given it another name. You did it first. They've got another name. Somebody over there has identified it. And then those names come together. They didn't realise that somebody else had identified it. That's often how it happens.
Are there any more questions? Is that one in the middle there? Yep.
You were saying something about clinical trials in relation to TCM. How are they adapted, these Western clinical trials, to this so very complex medicine system?
Yeah. I think the question is the complexity of the traditional Chinese medicines when they're taking into account the heat, etc. and so on. They use a different way of identifying the condition. I think that's what you're after. Is it? And then how we come with a westernized system. Is that what's behind your question?
Can a Western clinical trial actually be adapted, or is it possible to apply it to TCM?
Because
is it necessary?
Well, I think if we're going to look at it, if we're taking a westernized approach to the development, I think in traditional medicines you wouldn't always treat cardiovascular. You'd be treating other symptoms of the body. So in this particular case, they've decided cardiovascular is the area, so that's kind of already a westernized approach that's been taken with their specific targets that they're looking at. That is not traditional Chinese, so we're westernizing traditional Chinese medicines. Absolutely clear. But the Chinese are keen for doing that, and I think that's one of the kind of the opportunities. So we're not getting a traditional Chinese medicine into the westernized system. I think it's a westernized drug, and it's a different proportions as well. And because this particular concoction started out with about 12, I think they've got it down to about 6 plants. So they've decreased. And so they've decided they'll just go for cardiovascular, not all the other things that combination would have been used for, like affecting maybe the immune system, etc.
I think there's a question right at the back there.
I just wanted to clarify. You were saying earlier that you took the flowers in, I think it was Africa, and you took the soil with the flowers?
Yeah. The plants, yeah.
Plants, sorry. So that would make a difference on your crop. Yeah. So if you're taking those plants, so the plants, are they selecting the minerals or whatever they're taking from that soil? So if you took them to a different country, did you kind of test the soil for what minerals they had in them?
No, in that particular project, all we did on those few species, we did that work on. And in some plants, it didn't actually matter. The active compounds seem to be not dependent on the soil condition. So it's not true for all species. So, it's therefore knowing a little bit more about the biosynthetic pathway and therefore the importance of the chemistry of the soil in affecting the type of chemistry. And that's a whole research program, and that's why a lot of money is sometimes spent. Plectanthus, when it was being, the Catharanthus species for vincristin, vinplastin, a lot of time was spent on the agronomics and the soil conditions for that particular species to make sure it had a high level of the alkaloid, the vincristin, etc. So, each of these, if they were going to go into a westernised drug, there would be a big program behind them.
Are there any more final questions? Yeah, last one over there.
Yeah, just very quickly, I'm sort of do a little bit of work in Africa. And the interest there is in plants they could grow themselves and harvest their own drugs without having to go into bottles first or whatever. Is this research purely what you just said there in your last point of developing Western remedies from this knowledge, or could it actually lead into knowledge that could be transmitted to, say, poorer parts of the world so they could benefit from it directly?
So some of the traditional knowledge being transferred. Sorry, I want to make sure.
Okay, so could some of the output of the research lead to benefit to villages, basically being able to select more accurately which plants they could grow themselves to provide their own healthcare, basically free of charge at the point of use?
Yeah, very much. That's the work with the traditional healers in parts of Africa. That was not linked up with development of any westernised drug or something like that. That was very much a community-based project to make sure that those traditional healers had access to some of the plants that they were using to treat their patients. No pharmaceutical drug involved in that. You talk to the traditional healers, they're actually very keen to know how they can make money. They're actually keener than we were. Because, we went in as a conservation project and we said, no, we can't bring you X, Y, and Z. That's not the role. If you want to do that, you do that with some others. It was interesting because we actually ended up, and with one of those kind of plants, producing in a soap for treating some of the skin conditions. And that was help from local companies that could develop the soaps, not used traditionally like that, but you could see that maybe that was something that they could benefit. You'll often find that the communities are the one, and it's one person in that community, can see the dollar signs. So you go in there as kind of, this is a conservation project, actually, we're not interested in the drug, but if it works, they're interested. So, it's going back to one of the first slides I indicated on the Global Strategy of Plant Conservation. One of the key things that we're interested in is helping to conserve that traditional knowledge so it's there for use. And in Africa, that means trying to get that information in a written form because it's an oral tradition. In traditional Chinese medicine, it's a written tradition. Africa, it's oral. And we're losing it.
Okay, I think we need to wrap it up there. I think you can tell by the number of questions how much interest has been provoked by the talk. So I'd just really like to thank Professor Simmonds once again for the talk. I believe Professor Simmonds will be around for a little while having coffee, so if there are any other questions, then please don't hesitate to ask. Just to wrap up, then there is a feedback form which I would ask you to fill in, please. It's important feedback for the guys at the RSC, and then it only leaves me to wish everybody a safe journey home. Thanks.